Okay, so can someone tell me what was glyoxylate bypass? We discussed it yesterday. What was the purpose of doing that?
Sir, glyoxylate was further used for other amino acid and other molecules production.
Glyoxylate was used for that.
So oxaloacetate.
So can you tell me that how you can have one oxaloacetate for this purpose? Why are we doing this kind of a process? What was the purpose of that? Do you remember the pathway? Okay, you can see the reaction, right? Isocitrate lyase is splitting isocitrate to succinate and glyoxylate. What happens to that glyoxylate further? Yes. What happens to the glyoxylate?
It combines with acetyl-CoA,
Very good, and gives you what?
malate in presence of malate synthetase and water.
Malate synthase and water. Okay, great. Okay, and then what happens after malate is synthesized? Yes, Nikita.
So after that, it proceeds and it combines with uh, coenzyme A, I guess.
Malate.
So our main aim is to like produce one oxaloacetate extra so that we can make other byproducts such as we can go back basically. So after malate, one extra molecule of oxaloacetate will be formed, and after that we can go for that other process.
Okay, so you saw this reaction wherein isocitrate is being split to form succinate and glyoxylate. This glyoxylate will combine with another molecule of acetyl-CoA in presence of water, wherein we release CoASH. Malate synthase is your enzyme and you get malate. Malate, you know that is a part of TCA and can be converted to oxaloacetic acid. So the pathway used to look something like this, wherein you used to say that 2-acetyl-CoA will combine with 2 molecules of oxaloacetic acid, okay, and it is going to basically give you 2 molecules of citrate, 2 molecules of isocitrate. But out of these 2 molecules of isocitrate, one will go according to TCA. I told you oxoglutarate is nothing but ketoglutarate. So alpha Kg is same as alpha oxoglutarate. Okay, so one molecule of isocitrate follows the TCA pathway, while the another molecule is acted upon by isocitrate lyase. So that splits isocitrate to form a succinate and a glyoxylate. So you end up getting 2 succinate, one coming from the normal pathway, second by the isocitrate lyase reaction. This glyoxylate will combine with 1 acetyl-CoA and it will form a malate. So 2 malate any which ways are being formed by the TCA process itself. Now we have added 1 extra malate. So total you have 3 malate, 3 molecules of malate. These 3 molecules of malate can be converted into 3 molecules of oxaloacetic acid, 2 of which will continue in the cycle, but 1 extra oxaloacetate that is generated, you can use it for biosynthetic reactions. So you can use it for the purpose of synthesizing amino acids. You can also use it to go reverse of glycolysis, okay, to generate glucose molecules. So even that can be done, right? So that's why 1 oxaloacetic acid is going to be kept aside for anabolic reactions or for biosynthetic reactions. Okay, fine. So
Yes.
we also said that this is how it is shown here, wherein oxaloacetate can be converted to PEP. एनाप्लोरेटिक रिएक्शंस में हमने PEP कार्बोक्सी काइनेज के बारे में देखा था. जहां पर हम देख रहे थे कैसे PEP is converted to OA. It is also reversible. Once you get phosphoenol pyruvic acid, you can go back the scheme of glycolysis to generate hexoses. Okay, so for this purpose, glyoxylate bypass is very, very significant for biosynthetic reactions. Okay, so very quickly note these reactions down. Give heading is glyoxylate bypass, and let me know once you're done writing, so I will keep on changing the slides. हो गया लिख के? बदलू स्लाइड?
Yes, sir.
Yes, sir.
Yes, sir.
ये next जो slide है that I don't want you to write as of now. Okay, this you can refer from Gottschalk, because any which ways we will be focusing on this when we discuss gluconeogenesis, that is reverse glycolysis. Okay, so next unit में when we talk about biosynthetic processes for carbohydrates, that time we will discuss this part. ठीक है? So अभी ये मत लिखो, ये फटाक से अगर कॉपी कर सकोगे तो बेटर है. Very quickly. Okay, just note this down very quickly, and then we will move ahead to understanding the amphibolic nature of EMP and Krebs cycle. क्या बात हो गया लिख के? Great, amazing. I hope you are not just scribbling anything. Any which ways. Okay, chalo. So this part of glyoxylate bypass is something that you're going to read from Gottschalk. Okay, so please refer Gottschalk for this part. And now what we are left with is, if you see, we have completed till this part. Incomplete TCA tak we have finished everything. Now what we have to study is amphibolic role of glycolysis in TCA cycle and bioenergetics part. Okay, so let us first start with the amphibolic role of EMP and TCA cycle. So you already know the meaning of amphibolic. We have used this term several times now. You know that amphibolic basically means something which is both catabolic as well as anabolic, right? So intermediates which are produced either in the process of glycolysis or in the process of TCA cycle, they have specific utility in terms of biosynthesis processes, in terms of anabolic reactions. So every intermediate that you see either in glycolysis or in TCA is going to act as a precursor. It's going to act as a raw material. Like you can see right now, they have used short forms for intermediates of glycolysis. Like G6P is nothing but glucose 6-phosphate. So glucose 6-phosphate could be used as a monomer for building up polymers of glucose. Okay, like building up polysaccharides like glucose or cellulose or glycogen. This could be used as a monomer, wherein the phosphate group will be eventually removed and glucose will be added to the extending polymer. Glucose 6-phosphate can also be used to generate pentose phosphates. We have seen that in HMP pathway, right? We saw how in HMP pathway glucose 6-phosphate is converted up to the level of ribose 5-phosphate, a very important pentose sugar. Similarly, this could also be metabolized and used as a precursor for generating some aromatic amino acids. Fructose 6-phosphate used for generating amino sugars. Okay, you see DHAP, very important precursor for generating fatty acids, for generating several of the phosphoglycerides which are found in the cell membrane. So for phospholipid generation also, this is going to be a very significant molecule. 3-phosphoglyceric acid, 3-PGA, it is again a very important precursor or a raw material for synthesizing few amino acids like serine, glycine, cysteine. For these purposes, this is going to be very important. Okay. Other than that, if you look at PEPA, PEPA is a precursor for synthesizing aromatic amino acids. Muramic acid. Can you tell me where you have heard about muramic acid in the past? Do you remember this term, muramic acid?
N-acetylmuramic acid.
Very good. Where is it found, Saloni?
So cell wall.
Very good. Excellent. So it can also be used for generating muramic acid. Several lipids, amino acids, carboxylic acids, they can be metabolized to the level of PEPA, which means that PEPA can act as an entry point when lipid is your respiratory substrate, or amino acid is your respiratory substrate, or some kind of carboxylic acid is your respiratory substrate. So under those circumstances, they can be broken down to the level of phosphoenol pyruvic acid. From there, it enters your metabolic pathways. Is that very clear? It's not, see, every time, it's not essential that your respiratory substrate is going to be a carbohydrate, right? Many a times your respiratory substrate could be a protein, it could be a lipid, it could be a carboxylic acid. So then you have to first metabolize them to a level of an intermediate from which they can enter our central metabolic pathways. So for majority of them, the point where they, they reach after being broken down is acetyl-CoA. But for some, it could be even PEP, phosphoenol pyruvic acid. Okay, so it depends on what you are metabolizing. इसलिए अगर आप arrow का direction देखोगे in this particular case, you will see that it is pointing towards PEPA and not from PEPA. When the arrow is indicating from PEPA, it means that PEPA is acting as a raw material, acting as a precursor. But when the arrow is pointing towards PEPA, it's an indication that you can break these components to the level of phosphoenol pyruvic acid. Is that making sense to all of you?
Yes, sir.
Yes, sir.
Yes, sir.
Great. Now look at this. This may look very dangerous, but it is not. If you just pay attention, it is very simple. So if you look at the intermediates of TCA cycle, if you look at the intermediates of the Krebs cycle, each and every intermediate there has a significant role to play. You look at citrate. Citric acid is one of the precursors. It is one of the raw materials for synthesizing fatty acids, for synthesizing sterols. Okay. So various steroid hormones, citrate could be the precursor for that. Also, some of the lipid soluble vitamins could come from this. Okay. That's the reason why whenever you talk about several vitamin deficiencies, they also may indicate towards some of the imbalances in your respiratory metabolism. Okay. Other than that, if you talk about alpha Kg, I've already told you when we discussed TCA cycle itself that alpha Kg is responsible for synthesizing a very significant amino acid. What is that? Glutamic acid, glutamate. This glutamate is a very significant amino acid for a basic reason that if you have glutamate with you, by carrying out transamination reactions, you can synthesize other amino acids also, like glutamine, proline, arginine, because of which glutamate is given that kind of a significance. Because from that, you can synthesize the other naturally occurring amino acids. Also from glutamate, you can start the synthesis of nitrogenous bases, especially purines. Okay, so glutamate acts as a precursor for purine biosynthesis also, which you will see in the next semester when you study about catabolism and anabolism of nitrogenous bases of nucleic acids. Succinyl-CoA, again, very significant because it acts as a precursor for heme group, for porphyrin group, which constitutes the most important protein in our body, which we call as hemoglobin, right? You talk about oxaloacetic acid, lot of significance associated to it, because oxaloacetic acid can be converted to phosphoenol pyruvic acid. If you have phosphoenol pyruvic acid with you, you can go reverse of glycolysis, generate glucose molecules. You can also synthesize several amino acids from PEPA, like serine, glycine, cysteine, phenylalanine, tyrosine, tryptophan. These are all variety of amino acids that you can synthesize from PEPA. If you have oxaloacetate, you can also synthesize aspartic acid and asparagine. And asparagine is very significant because from them you can synthesize the other family of nitrogenous bases, which are pyrimidines. So can you see TCA is having such a huge importance even in terms of biosynthetic reactions, even in terms of biosynthesis or anabolism. And that's the reason why it is very apt to call both glycolysis as well as TCA amphibolic pathways. Many a times, people think that it's only Krebs cycle which is amphibolic, which is not true. Glycolysis is also amphibolic process. But in TCA, you see so much happening in terms of biosynthesis also, wherein every intermediate is possibly contributing to some or the other anabolic reactions. So TCA has gone, it has gained a lot of popularity in terms of its amphibolic nature, that it acts as both catabolic as well as an anabolic process. Okay. Now this part you will get to read as far as the EMP's amphibolic role is concerned. You will get to read it from David White. And TCA's amphibolic nature, you will get to read it from Leninger. So two separate books. For glycolysis amphibolic nature, please refer to David White. While for TCA's amphibolic nature, you will refer to Leninger. Okay. So please note that so that when you are reading it, you know which book to refer to. Okay. Now we will move towards the bioenergetics part. Okay, because I feel that it is essential to do bioenergetics. जब तक तुम्हारे दिमाग में the reactions are a little fresh, it is better to finish the bioenergetics part. So we will deal with that first. Okay, so let me cast my screen for that. As I told you, keep your glycolysis and TCA reactions ready because you will need them as I want you to do the calculations today. Okay. So what exactly is bioenergetics? So please understand that bioenergetics basically is a tally sheet. It's a tally sheet of, if for example someone is going to give you 1,000 rupees in your hand, especially our parents do this. You all would have experienced this in your school time, that whenever they give you some amount of money, they tell you that you have to give me accounts for it. Where did you spend it? How much did you spend for what? Right? So you maintain a tally of that how much money did you spend, on what did you spend. Okay, so similarly, when you have a bioenergetics tally sheet, what are you trying to do? You are trying to keep an account of where did you spend ATPs, how many ATPs did you spend, and where did you gain ATPs, where did you generate ATPs. So you try to make an entire tally sheet. Okay, now to make this tally sheet, you need to have an idea of what did you generate where. कहाँ पे मैंने ATP generate किया, कहाँ पे NADH या FADH2 molecule generate किया, and where did I spend ATP molecules. This has to be very clear in your mind, because until and unless you have a clarity of which reaction contributes in which manner, you will not be able to tabulate this. As you can see on the slide, they have very clearly written the reaction where I have either utilized an ATP or generated some kind of a product. Okay, so knowing that is possible only when you know the pathways in and out. Only when you know glycolysis and TCA cycle properly, you will be able to list the reactions down. Okay, so now you know the format, right? The format is going to be wherein one column is going to speak about the reaction. The second column is telling me how much ATP did I spend, did I earn, or how many NADH or FADH2 I generated, and how many number of ATPs are ultimately formed. Now, this third column will be helpful to calculate the ATPs that are coming from NADH or FADH2. Is that making sense to all of you? Okay. So now here I would want you to basically make such a table on your own, and then we will come back to this. Can you quickly do that on your own? 5 minutes. Can you do that?
For glycolysis only.
Yes, sir.
No, for glycolysis as well as Krebs, because now we are doing the complete bioenergetics.
Okay, sir.
We can do that also. If you want to write it simultaneously, we can do that. But you will have to give me the details. Chalega? Let's start from glycolysis. Can you tell me the very first step where you have either utilized or spent an ATP?
Glucose to
From
glucose.
glucose to
Glucose
glucose.
to glucose
Glucose to
6-phosphate.
Glucose
glucose
to
6
glucose
phosphate.
6-phosphate. Excellent. Great. How many ATPs did you spend?
1 ATP.
1.
One.
Very good. Okay, great. Next reaction.
Sir, glucose 6-phosphate to fructose 1,6 bisphosphate.
Beta, fructose 6-phosphate to fructose 1,6 bisphosphate, right?
हाँ सर.
हाँ. कितना ATP बनाया, खर्चा किया पहले तो?
Utilized.
Utilized. How many?
1.
1.
Great. Can you see next reaction where you have either generated, utilized ATP, NADH or FADH2?
From phosphoglycerate pgal to 1,3 bis phosphoglycerate.
3-phosphoglyceraldehyde to 1,3 bisphosphoglyceric acid. What did you do there?
Utilization of NAD to produce NADH.
How many NADH did you produce?
2.
2.
Very good. Remember this. You are doing the tally sheet according to 1 glucose. So all reactions are going to take place twice from this point of 3-pgal, right? So 2 NADH molecules are generated. Excellent. Can you tell me the next step? Anyone?
Production of 2 ATP from 1,3-bis-phosphoglyceric acid to 3-phosphoglyceric acid.
So 1,3 bisphosphoglyceric acid to 3-phosphoglyceric acid. You are generating an ATP. How many ATP molecules did you generate
Two.
there? 2.
2.
2
Great. Next step.
phosphoenol pyruvate acid to pyruvate
Phosphoenol pyruvic acid to pyruvate. What did you do there?
from ADP to ATP.
You generated ATP. How many ATP did you generate?
2.
2.
2
Very good. Now we are going to Krebs cycle. So do we directly jump to formation of citrate? No, right? There is something in between. What is that?
Acetylation.
Very good. Conversion of pyruvate to acetyl CoA. Can you tell me what did you do there? What did you generate in that process?
So we generate NADH.
How many NADH?
2.
2 NADH. Very good. Great. Next reaction in Krebs. Hello. Where do you have NADH in next reaction in Krebs?
From isocitrate to
Isocitrate to
oxalosuccinate.
oxalosuccinate.
Isocitrate to oxalosuccinate. How many NADH?
2.
2.
Very good. Next. Next reaction.
From alpha ketoglutarate to succinyl CoA.
Alpha Kg to succinyl CoA. So how many NADH again?
2.
2.
Very good. Then
Succinyl CoA to succinate.
succinyl CoA to succinic acid. What did you generate there?
The GTP.
GTP.
2 ATP basically.
GTP.
GTP बोलोगे या ATP बोलोगे?
ATP.
ATP.
फाइनल क्या बना?
ATP.
फाइनल ATP बना ना?
अच्छा फाइनल
ATP.
ATP.
हाँ, तो ATP बना तो कितना ATP बना?
तो 2 ATP.
2 ATP.
2.
वेरी गुड. 2 ATP बना. एक्सेलेंट. फिर
FADH to production from succinate to fumarate.
सक्सिनेट टू फ्यूमरेट. 2 FADH जनरेट किया हमने. एक्सेलेंट. फिर
फिर malate
So
to
from
Malate
malate
to
to
oxaloacetic
oxaloacetic
acid.
acid.
फाइनल स्टेप राइट. मैलेट टू
Yes.
ऑक्सैलोएसिटिक एसिड जहां पे हमने NADH जनरेट किया. अगेन 2 NADH. बराबर. So now if you look at the table, you will see this is how your table is going to look like. Okay? This is going to be the exact tally sheet that we generate. Now we have to convert NADH to corresponding number of ATP. That is very, very important. Okay? So I've told you earlier, how do you do this calculation? इसके लिए थोड़ा इलेक्ट्रॉन ट्रांसपोर्ट चेन का बेसिक नॉलेज होना बहुत एसेंशियल है. ठीक है? Please understand that when you are converting an NADH molecule, when you are oxidizing an NADH molecule to NAD, when this conversion is taking place in electron transport chain, a total of 10 protons are pumped from the matrix of the mitochondria. What do you mean by matrix? The internal environment of mitochondria. So you're pumping 10 protons from the matrix towards the inner mitochondrial membrane. टोटल 10 प्रोटॉन हम पंपिंग कर रहे हैं. ठीक है? नाउ 1 ATP जनरेट करने के लिए it is essential that we bring 4 protons back. Okay? Now let me tell you what I am trying to describe. Let me cast the screen of the whiteboard because I know that you have not done ETCS yet, as it's in the second unit. So I don't know if Niranjan sir has reached till that point. I'm sure that he might be dealing with membrane transport at this point. So I will just cast the whiteboard and try to explain it to you. Just give me a minute. Okay? Okay, so the tab is taking some time as it was switched off. Okay. Yes. Okay. So I'm sure that you all are able to see the whiteboard now. So when you talk about electron transport chain takes place on the inner membrane of mitochondria. I'm going to write it as IM. Okay? And this is the outer membrane, which I'm going to write as OM. This part is cytoplasm or cytosol as you call it. And this is going to be matrix. Now your ETC complex is ideally localized on the inner membrane of mitochondria. Okay? And when you are converting NADH to NAD in this process, you are going to channelize some protons out. So when you convert one NADH to NAD, you are channelizing a total of 10 protons. Okay, a total of 10 protons is thrown out. How? You will see that when you learn ETC in detail. So I'm not getting into that right now. And a part of this ETC is going to be the ATPase complex. Now, ATPase complex is an ATP generating machine. These are the complex जिनको तुम lower classes में F1 particle के नाम से जानते थे. Do you remember this name, F1 particle?
Yes, sir.
So this particular complex, what it does is, it is going to add a phosphate group to ADP and generate ATP. Now, how does this happen? When you're carrying out ETC and you're pumping out a lot of protons into the intermembrane space of mitochondria, you are generating a PMF. What is PMF? Proton motive force. Why are you generating a PMF? Because proton gradient intermembrane space में बहुत बढ़ रहा है, यानी बाहर पॉजिटिव चार्ज बढ़ते जा रहा है और मैट्रिक्स के अंदर नेगेटिव चार्ज बढ़ते जा रहा है. डू यू ऑल अग्री टिल दिस पॉइंट?
Yes, sir.
Yes, sir.
All of you.
Yes, sir.
Yes, sir.
Yes, sir.
What am I giving rise to? Because of this charge difference, I am giving rise to a membrane potential. Because of the charge difference. Okay, so this is giving rise to a proton motive force. Now, this proton motive force has a lot of energy stored in it, students. If I want to harness the energy in this proton motive force, I need to break it. जो PMF establish हुआ है, उस PMF को मुझे de-establish करना पड़ेगा. De-establish कैसे होगा? When I take these protons back, when I revert the charge, I will end up de-establishing the PMF, क्योंकि अभी charge based difference है ही नहीं. और अगर charge based difference नहीं है, then it is obvious, then I do not have this PMF. Are you understanding this point? Why am I, see, the whole process of ETC taking place is, I want to generate a charge based difference across the membrane. I want to generate a membrane potential. Because when that membrane potential is generated because of a proton, which we call as proton motive force, it has a lot of energy stored in it. I want to harness that energy. How will I harness that energy? By destabilizing the PMF, by breaking the PMF. Because when I break the PMF, the energy is set free. That energy can be used to generate ATP molecules. Now, how will I destabilize that PMF? I have to revert the charge. How will I revert the charge? Whatever proton was thrown out, take it inside. समझ में आ रहा है? अभी एक ATP generate करने के लिए by this ATPase complex, 4 प्रोटॉन को अंदर लेना जरूरी है. समझ में आ रहा है? अभी तुम बोलोगे ये क्या लॉजिक है सर, तो अभी मेरे तरफ ध्यान दो. Okay, I'm going to switch on the camera. to explain you how this F1 particle functions. ठीक है? तो ये तुम्हारा जो F1 कॉम्प्लेक्स है, लेट अस से दैट दिस इज F1 कॉम्प्लेक्स. ठीक है? F1 कॉम्प्लेक्स का दो पार्ट होता है. एक ये जो मेरा मुट्ठी है वो, ठीक है, दिस पार्ट एंड दिस पार्ट, ये मेरा F1 कॉम्प्लेक्स है. ठीक है? अभी ये जो पार्ट है F1 कॉम्प्लेक्स का, ये घूम सकता है. दिस कैन रोटेट. ओके? अब यहां पर F1 कॉम्प्लेक्स के इस हिस्से पर ADP अटैच्ड है, एडिनोसिन डाइफॉस्फेट. ओके? और इस हिस्से पर फॉस्फेट ग्रुप अटैच्ड है. मुझे अभी अगर ADP और फॉस्फेट को जोड़ना है, आई हैव टू गेट दिस फॉस्फेट ओरिएंटेड टुवर्ड्स ADP so that the terminal phosphate bond can be formed and ATP can be formed. Okay? For this machine has to rotate. For this to propel, it needs 4 protons to be moved in. When 4 protons move through it, this machine will propel to face the phosphate and ADP close to each other, forming an ATP. Are you understanding this part?
Yes, sir.
Yes, sir.
Yes, sir. Yes, sir.
Okay. So मैंने कितना प्रोटॉन बाहर फेंका था एक NADH के दौरान? 10. बराबर? और मुझे कितना प्रोटॉन अंदर लेना पड़ेगा एक ATP बनाने के लिए? 4. तो अगर मुझे ये कैलकुलेट करना पड़ा कि मैं एक NADH के बदौलत कितने ATP बना सकता हूं, इज इट डिफिकल्ट नाउ? नो, because I know that to synthesize 1 ATP, I need 4 protons to be taken in. I have thrown out 10. So 10 divided by 4 gives me the value of the total number of ATPs that I can generate from 1 NADH molecule. Is that making sense?
Yes, sir.
Yes, sir.
Yes,
Yes,
sir.
sir.
Yes, sir.
10 divided by
Yes, sir.
4. What is 10 divided by 4?
2.5.
2.5.
2.5.
So similarly, 1 FADH2, when converted to FAD, you are channelizing 6 protons out. And you will see how there is a difference in this proton when you do the ETC process. क्यों NADH के वक्त 10 जाता है और FADH2 के वक्त 6 ही जाता है, ये तुम देखोगे when you do ETC. So 6 protons are being moved out when you are converting FADH2 to FAD. And you know that to form 1 ATP, you need 4 protons to be taken in. So 6 divided by 4, how much ATP is it going to give you? 1.5. Clear?
Yes, sir.
Yes, sir.
Yes.
Yes, sir.
Yes, sir.
अब तुम वापस उस चार्ट पे जाओगे. अब ये चार्ट में तुम जाओगे तो जहां जहां पर NADH है, I know 1 NADH accounts for 2.5 ATP. So 2 NADH will account for how much? 5. 1 FADH2 accounts for 1.5 ATP. So 2 FADH2 molecules will account for how much? 3. Right? But you should have a question. The question should arise at this point. Can you see the difference? They have written 3 or 5. अब ये झमेला क्या है? This should be the first question that you should be asking me. That, sir, why is this difference only here? I can see it nowhere else. I can see it only at this point. And look at the reaction that this is corresponding to. This is the NADH that is generated in glycolysis. Do you agree that in glycolysis there is only one step where you generate NADH? Do you agree to this?
Yes, sir.
Yes, sir.
Yes, sir.
Yes, sir.
Where is glycolysis taking place? Where is glycolysis taking place? Yes?
Sir, in the cytoplasm.
Others?
The
Is option
cytoplasm.
correct?
Yes, sir.
Cytoplasm.
Yes, sir.
Yes, sir.
Cytoplasm.
In the cytoplasm of the cell.
Yes, sir. Cytoplasm.
Where is ETC taking place?
In mitochondria.
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 In the mitochondria.
Membrane, the membrane.
Membrane.
Which membrane?
The inner membrane.
Inner.
Inner membrane.
Very good. which means that if this NADH generated in glycolysis needs to participate in ETC, where will it have to go?
To inner membrane.
Excellent. It will have to cross the membranes of mitochondria, right? Because it is taking place on the inner side of the inner membrane of mitochondria. You can see where I have shown you the conversion of NADH to NAD. It is on the matrix end of the inner membrane of mitochondria. Barabar? So, bichara NADH, which is generated in the cytoplasm during glycolysis, has to cross these two hurdles. Which are those two hurdles? Outer membrane and inner membrane of mitochondria. Right? Now, in this crossing business, it cannot go by itself into the matrix. NADH को permit नहीं है membrane cross करके mitochondria के अंदर जाने का. So it has to depend on a shuttle system. It is like I have to send a parcel, but I cannot get inside the society. So I am giving that parcel to the watchman. Now, depending on how efficient the watchman is, whether he delivers the parcel immediately or he delays it, on the basis of that, the number of ATPs are changing. So the shuttle system, which is delivering the component to ETC, will decide whether it will be an energy efficient process or am I going to lose some energy in the process. Now, what is that shuttle system? That we will see tomorrow. We will not get into it today. And then we will do the totaling. Okay? But I wanted to do one task. Maybe not immediately tomorrow, but by next Tuesday. The task is going to be to do the same tally sheet if instead of glycolysis, I am using ED pathway. Some organisms do that, right? I will use ED pathway and then I go into Krebs cycle and ETC. So if my process is ED, TCA and ETC, then what would be the number of ATPs? So you will form a similar table for that. Will you do that by next Tuesday?
Yes, sir.
Yes.
Yes, sir.
Yes, sir.
That will be your task after understanding how we do this calculation tomorrow. You will do the calculation for that part by next Tuesday and tell me the results. And I have those calculations ready. I will show you the slide and we will verify it. Okay? That's it for today. I won't stretch 50 minutes. We are very close to 3:50. So we will stop at this point and we will continue tomorrow. Any queries till this point, please feel free to ask. Or else I hope that you have gathered what we have discussed today. Please ask if there are questions. Don't go back with the questions.
Sir, in the table, there is written number of ATP or reduced coenzyme directly formed.
Yes, because NADH and FADH2 are the reduced coenzymes, right?
Okay, sir.
It is either SLP that you note there, जहाँ पे हम direct ATP produce करते हैं by substrate level phosphorylation, or you are accounting for NADH and FADH2. NADH and FADH2 क्या है? They are reduced coenzymes, no? Because NAD and FAD are coenzymes. You have reduced them. That's why we call them as reduced coenzymes. Nikita, I want you to quickly tell me who is not here because just
Yes, sir.
wait, let me see if the attendance recorder is recorded.
Sir, Harshali is not here.
Nikita, it has recorded it. Great.
Okay, sir. That's
Correct.
it.
Thank you. Anything else? Anyone?